Mic test, mic test. Hello, good afternoon. Marami pa pong silya dito sa may unahan. Kindly occupy the front seat. So, we'll start now. Ready na? Magre-ready na. So, everybody in? Okay. Again, a pleasant good afternoon to each and everyone. Actually, this will be the last session for the different fora for this NSTW. And at this point, I would like to welcome all our participants here, yung interesado sa coconut. And actually, the technology that we are going to share with you ay isa sa major intervention which we think will really help the coconut industry. I know sa iba't ibang lugar kayo nanggaling, and there are representatives from DOST regional offices, from the different SUCs, UPLB, RTU, kasi nandun yung aking hipag, and then UPMIN, BUCAP, ano pa, and PCA. And I would like to recognize yung ating mga pillars sa biotechnology, because this is actually a product of biotechnology. We have with us Dr. Ben Pecson. Sir, tayo naman. And of course, Dr. Rita Laude. And Dr. Antonio Lalusin. Laurena, I'm sorry. So, gising kayo, gising. And of course, andito rin ang aking dating professor, si Dr. Maria Cedo. Siya yung another kilalang-kilala sa tissue culture. Ano yan? And then Dr. Olive Damasco. Okay. And Dr. Annabel Nubero from UPMIN. Okay. And Dr. Berina Exconde from Biotech. Baka ako yung mapagalitan. Sino pa? Dr. Marisa Estrella from Bicol University. Okay. So at this point, si Rose Reglos. Sarah. Okay. So, siya yung hindi naman nanay ng tissue culture, ng coconut somatic embryogenesis. Pero isi-share niya kung paano na-develop ito at kung saan nanggaling. So, our speaker is Miss pa po yan, Cristeta A. Cueto. She graduated her Bachelor of Science in Agriculture and Master of Science in Plant Pathology ang kanyang major. And he finished his doctoral degree as major in genetics and minor in molecular biology and biotechnology. So, siya po yung mag-share sa atin ng technology ng coconut somatic embryogenesis. And may I call in our speaker?
Thank you, Tita Ed. Hi, good afternoon. Thank you sa PCAARRD for inviting me here. Tapos, masaya kasi parang reunion. I see old friends. Nandyan si Ma'am Laude, si Dr. Pecson, yung si set group, nandyan. Tapos ay yung mga kaibigan ko sa LB. Parang ang reunion. Ayun. Thank you for coming. So, good afternoon. We would like to share with you yung updates natin on coconut somatic embryogenesis. Coconut, the tree of life, Cocos nocifera, we all know this, and it has been noted as the tree of life not only for its uses as food up to shelter, but its function to nourish, yung role niya for the nourishment of the coconut industry. In the Philippines, 68 out of the 79 provinces covering 3.56 million hectares are devoted to coconut farms. And these farms provide livelihood to, just this morning we were informed, 2.2 million of the coconut farmers, no longer 3.5. These 2.2 million farmers are composed of landowners, yung farm workers, and tenants. And in terms of hectareage, the Philippines ranks second in the world next to Indonesia. And in terms of copra production, it ranks ranked third next to India and Indonesia. In terms of the contribution to the economy, the coconut is the fourth largest contributor to GBA in agriculture, and copra remains to be the major export product. Considering the importance of coconut in us, there is a continuous need and demand for quality planting materials. Coconut seedlings, which are early and high yielding, with resistance to pests and diseases, tolerance to abiotic and abiotic stresses, and could be good source of high quality oil and cocoa sugar. Yes, there has been a demand, continuous demand actually, for quality coconut planting materials, not only for coastal zone expansion, but as well as for replanting purposes. We need to replant the coconuts to replace the old and senile palms and those infected with CSI, the one with badly hit the Calabarzon areas in the last 5 years. And then we still have this kadang-kadang infestation in Bicol region, although at a lower scale, but still the disease is with us, and we need to cut and burn, and then the need to replace. And then we also have these coconut farms which were stricken by natural calamities like typhoon. More so, there is the need or the demand for raw materials for high-value coconut products like virgin coconut oil, cocoa diesel, cocoa water, and cocoa sugar among them. And then for the galactumanan from Makapuno is also gaining popularity in both food and non-food industry. So, as we know, there is this increasing demand for high-quality planting materials. But where are we now? For our planting and replanting program, we are still dependent on the mass production of open pollinated seed nuts, which are grown in seedbed and coconut seed garden. And then we are also into this hybridization program, which takes around 15 years to be able to develop a hybrid. It takes a long time, but still, this is a better option of mass-producing quality planting materials. And with these two technologies, the use of open pollinated and the hybridization technique, we are producing one seedling from one nut. Another existing technology to mass-produce the coconut is the embryo culture technology, which was developed by the late Dr. Emerita de Guzman of UPLB. Primarily, it was aimed to rescue the Makapuno embryo. But then, with the developments, it was used to other purposes, like in cryopreservation work, germplasm exchange, and in vitro screening for pests and diseases and other biotic stresses. And the Makapuno program of PCAARRD, actually this was PCAARRD-funded program, implemented at ARC, but then was able to establish satellite laboratories in different parts of the countries. We have one in Zamboanga, Davao, PCA Tacloban, and PCA Pangasinan, and Cavite State too. With the program, we were able to equip those satellite laboratories and then establish demo farms side by side with the laboratories. And in case of ARC, we were able to have a 4-hectare Makapuno farm, and during the 20 years of harvesting, we have the 80% makapuno yield obtained from these embryo cultured palms. So, just to say na this mass production of coconut through seed nuts and embryo culture produced only one seedling from one nut or from one embryo. Hence, there is a need for a mass propagation system. We need to develop one to support the planting and replanting program to replace these senile and infested palms. And so, coconut somatic embryogenesis, this is the main topic that we will be presenting. It offers a promising alternative to mass propagate the coconut using immature flowers, plumules, and immature embryos. These 3 explants were found to be responsive in terms of plantlet regeneration. And from one explant, we expect to produce hundreds or thousands of plantlets. We could say that we were saying coconut is a recalcitrant crop. It can just germinate easily with or without soil. You could just see it germinating. However, when it comes to coconut somatic embryogenesis, it proved otherwise. Coconut somatic embryogenesis technology faces challenges. It needs well-equipped laboratory, not only in terms of equipment, but the laboratory itself has to be supplied with, we should have a steady supply of electricity and water. Otherwise, even if we have this high-tech equipment, or even not high-tech, the basic equipment, if we do not have the steady supply of electricity, we will be facing some problems. And then it requires highly trained and skilled technicians, not only the skills, but the passion to do coconut somatic embryogenesis. To do tissue culture work, actually, this really requires patience and commitment. And it has been observed that coconut cultivars have varying response in culture. Recalcitrant, as we were saying, they could just germinate easily, but when we culture them, we could see that they don't respond all at the same time, and they are not giving the same response at one time. There are cultivars which do not form colloids at all, and there are plumules which are really, really responsive. And then it entails a long incubation period to mass propagate coconut planting materials through coconut somatic embryogenesis. Say, for example, for the mass production of coconut using open pollinated and hybridized nuts, we could, or even embryo culture, we can get one seedling in 9 months period. However, with coconut somatic embryogenesis, we could get plantlets ready for planting in the soil as early as 4.5 to 5 years after initial culture. And then the next 4 slides will show us a summary of the extent of the work carried out in different coconut tissue culture laboratories worldwide. As we could see, research on coconut started in 1976, wherein the media that we are using now, the Y3 medium, was formulated by UNs. And as we can see, there are only few explants that were able to regenerate plantlets. We have immature flowers, plumules, and leaves. But in terms of leaves, we could hardly repeat the regeneration, plantlet regeneration using these explants. Immature flowers and plumules were found to be more responsive. And between plumule and inflorescence, plumule is more responsive than inflorescence. So we could see here the different response of the different tissues. There are cases that immature flowers just form colloids, but in some cases they could regenerate until plantlets. In case of the Philippines, we were able to regenerate plantlets from plumules, immature embryos, and immature flowers. However, only those from immature flowers and plumules were able to go to the field. This slide tells us the modification of the culture medium that has been carried out to be able to come up with the working protocol for coconut somatic embryogenesis. So these are the different researches carried out for the culture medium suited for coconut somatic embryogenesis. And we could see that it is more of the interplay of the growth regulators, auxins, and cytokinins, 2,4-D in particular. So for plumules, plumule explants, again, we could see that aside from the growth regulators, the effect of charcoal and other growth regulators as homobracinoli, GA3, have an effect on the response of the plumular tissues in culture. For the inflorescence, just the same, it is an interplay of the different auxin and cytokinin and GA3 to be able to obtain the different stages of culture, inflorescence. So PCA, the coconut somatic embryogenesis, is now on its 28 years in the making. We started the project working on coconut tissue culture in 1989 under the GTZ project, followed by the ECSTDT program, and then we have funding from the DOST-PCAAARRD. ASHAR came in, and then we have DA Biotech from 2007 to 2010, and now we are implementing again this program. This is ongoing, reinvigorating the Philippine coconut industry through the coconut somatic embryogenesis technology, a DOST-PCAAARRD funded. We will discuss about this project later. So the 28 years in the making involved the inputs of the PCA tissue culture team then, which was headed by Mrs. Erlinda Pirillo. She was the division chief of tissue culture division and retired from PCA in 2008. And we have some consultants. We have resident consultant then from GTZ, Dr. Ebert, and then we have these collaborators, partners who have been working with us in the development of these coconut tissue culture works. And for PCA, we have Maria Bilen Paloma. She has resigned from PCA in 1991. Actually, I replaced her position then. And then we have Boots. She is the only one remaining with us in tissue culture. The two of us are now the permanent staff working on tissue culture. And we have Osmond. He retired also, joined a private company, and then myself. And this group has been backed up by a group of men and women which were hired under job order or contract of service. And some of them are still working with us under the project. Ayun. So, as I have mentioned, PCAARRD, coconut somatic embryogenesis, is a work in progress. So, this is the technology that we will be presenting. This all started with the collection of explants. For immature inflorescence, a technique has been developed by Rillo in 1989 to collect the immature inflorescence. Actually, it is in inflorescence number 4. You could imagine the coconut palm, the spur lip is leaf 0, and you count it, you just follow the phyllotaxy. The leaf number 4, under the leaf number 4, embedded is inflorescence number 4. So you could imagine how we how destructive it is to collect the inflorescence, but it has to be non-lethal. You have to keep the mother palm alive. So for immature leaf, Booth was able to develop a technique to collect immature leaf without killing the palm. So since 1989, we have been working on immature flowers, immature embryos and leaves. And then when the EASY project came in, we started working on plumule. So these were the original group, Rillo, Cueto, Orense, Ubaldo, and Ebert, who had started the work in coconut tissue culture at PCA. So for plumules, by the way, we are using mature nuts, which are not yet, not the brown one, but in the color break stage, 10 to 11 months old actually. So these are the explants. Our sources of plumule, this is a 10 to 11 month old zygotic embryo, and then we have here the inflorescence and immature immature embryos. The meristem here, the plumule, and rachila here are sliced into small pieces. Subject them in culture medium with a combination of 2,4-D. 2,4-D in this case is higher in concentration than the ones in the regeneration phase. So you allow the tissues here to form colloids, and these colloids, you allow them to multiply until they become embryogenic. And once they have the potential for embryogenicity, they will be transformed into somatic embryos. And these somatic embryos will then develop into plantlets, something like this. For plumule, it will take around as early as second month to have them initiate colloids. But for inflorescence, it will take at least 4 months. Sometimes it will take 6 months before you could see some colloid formation. But between immature embryos and plumule, immature embryos is still less responsive in terms of colloid formation than plumular tissues. So once we have these colloids, we just keep on multiplying them, subculturing them in fresh medium with with increasing levels of 2,4-D until we have enough colloids that will give us somatic embryos. So by the way, alam naman natin yung somatic and zygotic, no? Zygotic is the product of fertilization. Ang somatic embryos, bali ay coming from somatic tissues yan. Wala siyang vascular connection from the mother, sa mother pump, sa mother tissues natin, no? Ayun. So, yung multiplication itself, it will start on the 4th month, and then it could be continuously multiplying. And then for embryogenesis, this is the start of the regeneration phase. It will start on as early as the 7th month if we have responsive cultures. But sometimes it will take as early as late as 23rd month before we could have somatic embryos. So based on the early responding somatic embryos, we could have them germinated on the 12th month and then ready for ex vitro establishment on the 3.5 years after regeneration. And now at ARC, as I was mentioning earlier, we have field planted plumule and inflorescence-derived coconuts at ARC, and some of them are already at the bearing stage. This shows that the protocol is technically feasible, wherein we could have the nuts or the flowers, they form colloids, they form somatic embryos, they regenerate into plantlets, field planted, and now we can harvest again. No? We have nuts harvested. They are either subjected to embryo culture or to seedbed for mass production purpose. So based on what we have done, we were able to conceptualize this pathway, which will tell us about the development of the cultures in vitro, the inflorescence and plumule-derived cultures. And this is based on the morphology of the cultures. This will tell us which type of culture will go to the next stage or to the next medium to be able to have these plantlets, or which type of culture will go to the bin at a specific stage of development. Ayan, this one is helpful for us in determining the quality of cultures that we are maintaining. And PCA has been supporting us as well in the development of this protocol, and we are continuing our studies with immature flowers, immature leaves, immature embryos, and plumules. The plumules that we are using are from the coconut varieties, which are not being used under the DOST-CCAARRD program. And also in this study, we are working on the secondary somatic embryogenesis pathway to increase the regeneration of plantlets that we are getting. With the primary cycle, we were getting only as much as 120 plantlets. That was the highest that we got. And for secondary somatic embryogenesis, we were getting then 235 plantlets per somatic embryo for one cycle. But as you could see, CSET protocol is easier said. We have here many pitfalls. At different stages, as early as the colloid initiation phase, we experience browning. With brown tissues, what do you expect? They go to the bin. And then at the regeneration stage, we will experience the hostorial formation, the early root formation. These are not good quality cultures for us. So indeed, yung CSET has been proven difficult, and there is where the challenge is. Continue to face the challenge. And another thing that we are doing, we have just started this one. Actually, the cell suspension we have not started, but we are now working on anther culture. And based on earlier trials, we have identified the anther response, the age of anthers that is responsive for culture. And we have now colloids and somatic embryos derived from anthers that are being maintained in the lab. So this is the program that we are now implementing under the fund provided to us by the DOST. This is a 215 million project, a 5-year program actually, which started in 2014, and we hope to finish in 2019. We have here the implementing agencies. We have UPMIN, care of Dr. Anna, UPLB, Dr. Cedo, Ms. Rose, Dr. G, Dr. Oli Damasco, Visayas State U, and then Bicol University, Dr. Maris is with us. And of course, PCA, we have PCA Zamboanga, we have Mr. Emmanuel and Harriet with us, and ARC. So this project aims to provide the planting materials, CSET-derived planting materials to different regions of the country. So they are strategically located actually. We have Region 4, UPLB, Region 5, BU and ARC, Region 8, BSU, Region 11, UPMIN, and Region 9, PCA Zamboanga Research Center. So, these are the provinces that will be catered by the specific project in terms of plantlet distribution. We hope to have plantlets by end of year 5. Which province first? That is yet to be seen. Anyway, so the objectives of the program actually is to acquire knowledge on CICIS, that is a research center in Mexico. We hope to acquire the knowledge on CICIS protocol for in vitro culture of coconut using somatic embryogenesis technology. It has been known that the CICIS protocol could produce 10,000 plantlets from one plumule. If you will compare, we are just getting around 120 or plus the secondary 235, that is around 350 plantlets compared to 10,000 plantlets from one plumule. We are really far, far behind. But unfortunately, we weren't able to get the CC Mexico, so we just settled with the PCAARRD protocol. Anyway, so for the second objective of the program, we have the capability building of laboratory personnel and upgrading of tissue culture lab for mass production of planting materials. This has been accomplished during the first year of the project implementation. We have labs in UPB, Bukap and Zamboanga. They have the big labs for the project. And then ARC, coming soon, yung extension. And then UPLB and UPMIN and Bisaya, BSU, they are requiring more space for their culture rooms. And to produce quality planting materials through CISET in order to augment the replacement of typhoon-damaged palms, CSI-infested, and old or senile palms, and for the expansion in coastal areas. So these are just the list of the varieties that we are using. Laguna tall. For the talls, we have Laguna, Baybay, Bago Zero, and Tagnanan. Tagnanan is one, they are all parentals being used for the hybridization program. And for the Tacuna, for the dwarf, we have Tacunan Green Dwarf, Gala, San Isidro. San Isidro Dwarf was the first, the variety of the first inflorescence-derived clone that we have field planted. And then we also have Malayan Red Dwarf for the hybrids. And we have new additionals, the Katigan by Laguna and Tacunan by Tagnanan hybrids. So PCA Zamboanga maintains the germplasm collection. That's why we are getting the embryos from PCA Zamboanga Research Center. The group of Harriet and Estong are preparing the embryos for shipment to these implementing labs. And once they have reached the lab, they are prepared, they are surface sterilized until ready for plumule excision and inoculation in UN's basal medium. So, as of now, most of us are in this stage, yung colloid and somatic embryogenic phases. We are maintaining, we have initiated 106,000 plumules and now maintaining 163 colloids. And for somatic embryos, we have 16,273 embryos at different stages of development that are being maintained in these laboratories. And we have 963, this is a combination of the shootlets and plantlets that are being maintained in the lab. We have actually 100 plantlets already, so 863 shootlets are available. And we hope all these shootlets will be converted, will further develop into plantlets. What does this slide say? This slide shows that the protocol could be repeated. This is the regeneration is not just happening at PCA Albay Research Center. We have plantlet regeneration from UPLB and then Bukap. We are maintaining already some plantlets, and UPLB has exceeded the success rate that we have obtained, they are getting 200 shootlets from one plumule, whereas in our case, we started at 120 plantlets per plumule. So at least we could say, and we are happy that the protocol can be transferred, it's repeatable, and we hope that we will be getting more of the responsive plumules before the program ends in 2019. So these are the CSET laboratories that have been upgraded. We have one in UPLB. Actually, there are 3 labs involved in UPLB. We have one in biotech, in crop science cluster, and in the Institute of Plant Breeding. And then at ARC, BUCAF, VSU, UPMIN, and Zamboanga. If those who have been to Zamboanga, the COCOFED building has been converted into a CSET laboratory. So you could imagine how big the coconut tissue culture lab in Zamboanga Research Center is. So these are samples of the colloids that we are getting. We want this type, the globular, shiny type, pearly white colloids, because they have more potential to be embryogenic. So we are getting colloids not only from the talls, but we also have from the dwarfs and the hybrids. We just hope that these colloids that we are maintaining, the 106,000 colloids in the lab, would become embryogenic and eventually become somatic embryos and eventually plantlets. These are some of the somatic embryos that we are getting. Some have started to germinate. We just need them they just need some hardening before we could transfer them under lighted conditions. And then we have some shootlets, San Isidro dwarf, Baybay, Malayan red dwarf by Tagnanan, Takunan dwarf by Tagnanan tall. These are some of the shootlets that we are maintaining, and then the plantlets. Unfortunately, the plantlets are not yet ready for ex vitro establishment. They are still at the hardening stage inside the laboratory. These are plantlets coming from the lab of UPLB and ARC. So where do we go from here? There is still a lot of things to do to be able to improve the protocol. As you could see, it takes 5 years before we could have plantlets ready. How we wish we could shorten it so that we could make the seedlings or the plantlets available to meet the demand for quality planting materials. We are sourcing embryos from selected palms from the recommended varieties of PCA. Thus, we could say that we are producing, mass propagating quality planting materials. And we need a working protocol. Though we could say that we could repeat the protocol, we could get plantlets, we still need to refine the protocol to reduce the cost. We could see it entails a lot of costing for the labor, for chemicals. Imagine the long incubation period. How much chemicals, labor, cost of electricity have been devoted to produce these plantlets if they will be taking such a long time. And then we look forward for the application of the CSET technology for mass propagation, yung real mass propagation, wherein we will not just be settling for 10 plantlets per plumule, but as we were discussing, 200 plantlets per plumule are good enough at the start. But with secondary somatic embryogenesis, there's still a lot to hope for. So, clonal propagation by somatic embryogenesis, we hope will initially supplement and may later replace the conventional method, the seedbed, the hybridization, and the embryo culture technique of mass-producing coconut planting materials. The hybridization, of course, it cannot be replaced by mass propagation because what we will be mass-producing here would be the F1. So what would be used in the hybridization project is the mass propagation of the F1s that we will be getting from the hybridization work, so that the nuts will not just be giving one plantlet, but the F1 nut will be producing more plantlets if this protocol will be used. So eventually, it will ensure massive planting in the country and increase the supply of raw materials that are being demanded from us for the high value-added coconut products. And we hope that we could improve the lives of our coconut farmers, and then we could provide enough supply of raw materials for our processors and for traders, increase volume and trades, and for the government to increase its dollar earnings. So indirectly, kasi we will be producing more, we will be earning more. So, iba na. Ayun. That's the end of the talk. Thank you very much. May acknowledgement. Ayun. Ayun.
Thank you very, very much for a very extensive explanation of the technology. And now the floor is open for your questions. Maybe this is the reason why Dr. Christy Cueto has remained single. Ito yung kanyang naging baby. All her life, tissue culture. And we are happy na kita na yung successes with the help of other collaborating agencies who has been part of the generation of this technology. Actually, talagang gusto sana namin yung sa Mexico, which is 10,000, but we were very unlucky to have it. And we are very happy that we have our local technology coming from our local researchers na from other countries. Actually, kung Mexico yung successful, parang susunod na dito ang Philippines actually, because we have attended several international conferences on somatic embryogenesis of coconut. Parang tayo na ang pangalawa. Next is India and Sri Lanka. Yes, sir. So, question, Dr. Ben Pexon.
Christy, if we have, 320 million coconut trees, and if we assume that 20% of them are senile, what will it take using this technology? How long will it take to produce 64 million plantlets? How long will it take? What will it cost? Because that information is needed by PCA. Ano ba talagang kailangan to produce that much? Actually, you probably need more than that because more than 20% of them are senile.
Naman po yun. Oh, yun. So, yung sa pagdating sa costing, sir, hindi pa namin talaga natitingnan with regard yung sa costing na mangyayari kasi nasa ano pa yung exploratory kumbaga. Though ang target natin makaproduce ng plantlet, pero kung kung susugtugin, talagang may mga refinements na pa along the way na nangyayari. Tapos, ang pagdating doon sa number o kung gaano katagal, gaano karami. Kasi sa ngayon, yung aming, ang na-initiate na yan, nasa 106 and we are just getting around 963. Pero ito naman ay regenerated from the year 2, yung year 2 na project implementation period. So, pero yung 64, napakalayo pa, sir. Napakalayo pa namin. Pero, estimate, sabi niya sa akin kahapon, happy na muna siya dun sa 200. Hindi, sir, Oo, yun, hindi ko masagot talaga, sa totoo lang, kasi napaka-variable ng response. Ang una man ang titingnan dyan, anong varieties ang itatanim natin. Kasi among the varieties that we are working on, 14 po yun actually. We have just found 5 responsive pagdating sa colloid formation. And then we have identified 3 responsive varieties pagdating sa somatic embryo formation. So, marahil yun ang isa nating titingnan. Anong varieties? So kung magsisiro in tayo dun sa responsive, Laguna is the most responsive among them. Kasi responsive siya sa colloid stage, responsive siya sa somatic embryo stage, and then sa plantlet regeneration, dun din nakakakuha ng mataas na plantlet regeneration. 64 million, yun ang kailangan nyo. Ganun kadami. Pwede sir, email ko na lang sa inyo. Aha. Kasi babalikan namin yung calculation, bali dun sa available data. Pero mahihirapan po talaga kami nun. 64 million. Pero masaya na rin po ako kahapon kasi I was talking with the administrator. For the first time kasi nakausap ko siya about the CSET. It just so happened that he is a friend of Dr. Concepcion. So medyo naano siya kasi may passion daw talaga siya for tissue culture. So I was telling him na we were getting 120 and now the highest is 200 sa UPLB nga na lab. And then Mexico is getting that much. Sabi niya, doon na lang muna tayo sa kung ano yan. Masaya na siya dyan. So masaya na rin kami. Pero kung ano naman sir, ang ano dito, what we can assure, nandun yung passion namin. We will not be settling for 200. Actually, nag-set kami ng 1,000, but we are trying to forget muna yung mga targets na 1,000, 10,000. Ang ano namin, ma-assure namin na we won't settle at 200, we won't settle at 300. Kung kaya naming abutin yung 1,000, 10,000, hopeful kami doon. And nandun yung tuloy-tuloy lang yung passion and commitment. Opo, yun ang maano namin. In 5 years time, sana with God's help, laging sabi namin, dasalan natin to kasi we can't do this alone.
Pag natin yun, pwede na tayo mag-retire, no? Okay, yes ma'am, Dr. Laude.
Actually, I know you reported only 10 plantlets regenerated, successfully regenerated from 70,000 colloids, no? But now you have regenerated already successfully from immature inflorescence and from plumule, yeah. So we are so thankful about that. And I would just like to ask, you said for seedling, no? It would take, from the colloid to seedling, it would take about 2.5 years. But that's only to the seedling stage, no? Minimum. So kung manghihingi sila ng seedling na yun yung 2.5 years from the laboratory to the field, di ba? To the nursery na. Oo. Yeah. So malaki ang cost from colloid to the yeah. Okay, so congratulations. So probably the costing you can start from building up the laboratory to the 200 plantlets for every 200 plantlets that you can get from each plumule, right? Pero wala namang problema sa explant, di ba? You can get Sambuanga.
Actually, yung sa, yung nasa program, pag nag-uusap-usap kami ng mga project leaders at saka yung staff, sa laki ng investment ng PCAARRD, 251M, kasama ang infra, equipment, capability building. Kailan kaya mababawi yon in terms ng plantlets na regenerated? Kasi pero kung tutusin natin, yun nga, malaki ang devoted din sa infra. Bali ay nasa tig pa 3M per lab noon. Yung
5M.
initial, no? 5M per lab.
We have spent 5 million per laboratory initial.
For the upgrading.
And then additional every year we have given. It's really a very expensive technology, ma'am.
But we already established some of the laboratories.
Yes, ma'am. Actually, in Mexico, they have a
A biofactory.
biofactory. Yeah. And when we were discussing this technology with them, what they wanted is they will be talking only with one laboratory. Ayaw nila na maraming laboratory ang mag-start. Kasi nga, medyo mahirap i-transfer ang technology and making sure of it, it's really very expensive. Yeah. Yeah, the skill, the passion, the tender loving care for the cultures, yun ang sinasabi nila. Any more other question? Yes, Dr. Laurena.
What is the difference between the cultivar that is used by Mexico and our own?
Sa ngayon po, ang ginagamit nila MYD, Malayan Yellow Dwarf, which they have found resistant sa lethal yellowing. Yun ang kanila na work on. Ay nagkataon, ang MYD, hindi naman siya kasama sa recommended varieties ng PCA. Actually, marami sila. Meron silang Mexico Tall, yung mga sa kanilang varieties ang ginagamit. Very responsive po, opo. Sa kanila, opo, meron silang mga identified na 10,000 plantlets ang nakukuha nila from one plumule. Yun po ang sinasabi ni Dr. Oropesa.
I remember when si Linda Rillo is still with PCA, binigyan ni Dr. Oropesa ng protocol si Linda. And Linda followed the protocol to the letter very religiously. And yet we cannot produce. Then probably they are not giving us everything.
Actually, ganun nga po yun. May industrial secret. May secret talaga. Kasi may mga publications, we have communicating with them, with Dr. Oropesa. Talagang, kung pwede lang suluhin, talagang one-on-one. He's trying to give us hints ng ganyan. Na yung mga sinasabi niya, ginagawa naman namin. And then yung mga protocols nila na publish, sinusunod. Pero hindi talaga namin maulit yung kanilang protocol. At hindi lang naman po kami. We have tried comparing the success rates of Sri Lanka and India. Kasi kaming tatlo bali yung gumagawa ng protocol nila, nag-adapt na sabay-sabay. Talagang we're sharing the same sentiment. Hindi namin maulit yung Mexico protocol. Ayun.
I was also thinking that that protocol was accidentally discovered by Mexico because I was expecting France to be the one who is going to solve the problem because of their extensive work on tissue culture. Palay ko natsambahan lang nila yun, and the technique is so simple, they don't want us to tell us.
Isa rin maganda, sir, sa group ng Mexico, talagang extensive yung ginawa nila. They did work from, meron silang researches sa anatomy, yung lahat, anatomy, biochemical, and then they have the molecular. Talagang very encompassing. Advanced na advanced sila ang work nila sa parang backup talaga ng multidisciplinary approach ang nangyari. Pero yung sa France, ganun pa rin, tuloy-tuloy pa rin. Pero ang Mexico and France, they are working together sa coconut pa rin. May mga projects pa rin sila na magkasama sila.
So under the Coconut Genomics Program, also funded by PCAARRD, we are going to have the genome of our coconut, the dwarf variety. And I hope that this will help you in understanding the somatic embryogenesis of coconut. Yung sa tall variety, matagal pa kasi heterozygote ang tall compared to the dwarf variety.
Ayun, magandang baliktayon. Pero yung sa ngayon, ang nagwo-work na lang talagang laboratory sa Mexico, ay sa tissue culture, we have Sri Lanka, India, tapos ay Mexico, Philippines. Kasi ang France, they are more of the yung research aspect. Pero doon sa meeting nga namin ni Dr. Edna sa Bali, nagulat kami na ang daming gustong magtrabaho sa coconut embryo culture, coconut somatic embryogenesis, ganyan. And then yung iba, akala namin, naka-take off na, yung pala, kahit embryo culture, mag-start pa rin lang sila. And they are still very interested with the Philippine makapuno. Ayun. Yun ang ano doon. Ang kanilang hinahanap. Philippine makapuno.
Yes, sir.
We say that we've spent 200 million pesos for this sort of thing. It looks high. On the other hand, so many years ago, copra was our #1 export. And unahan na tayo ng Indonesia, and we're third to India. When you ask for money from the gumagawa ng budget, di ba? The question to ask is, what is the cost of not investing more in R&D? Because I know very well that we are lacking researchers in the coconut sector. You yourself have you have so many positions which are not filled. The country really should spend so much percentage of its GNP for research. So when you ask for grants, not only you, but PCAARRD, show what is the cost of not acting. Now, overtake na tayo ng mga other countries. And if we say that agriculture must be modernized and we have to use these better technologies, point out na our percentage of money spent for R&D is small, and this is a technology where we used to be #1 and now we're slipping behind. Not only do we need to replace the senile we only have 5 na nagre-respond. We have 15 varieties. But you need various varieties for various locales, di ba? So you could use an argument, what is the cost of not funding more? Because we need more coconut scientists, we need more people knowledgeable in biotechnology, and say, hey, this is really high priority. Because the government says, responsive naman sa the needs of the people, di ba? Anyway, to help you in your funding.
Actually, this is the first time na nagpondo kami ng program this big for 5 years sa PCAARRD. And ang next dito ay yung aming malaking coconut genomics program, which is also 214. So we were quite lucky dahil yung secretary dati and the USEC is really pushing us to pursue this development ng technologies for coconut, which tama ka sir na huli nga tayo doon sa puntos na yun. But anyway, with this, we are already on the 3rd year. Parang 4th year na kasi kami. First year namin is yung development nga ng mga facilities, and then parang in 2 years, medyo nakakita na kami ng liwanag dun sa technology. And sabi nga niya, repeatable. So I think yung asking for more funds will be easier if we can show them that we have lots of plantlets and kailangan na namin ang maraming space for rearing more plantlets para sa ating replanting program. Any other more questions? Po? Yes, ma'am. Identify yourself, please.
Sophia Mahalino po, high school student. I just want to ask, if there is a difference between the quality of product that we get from the coconut tree that with technology and without. Apa.
So far, wala kaming nakitang difference. In fact, yung field plant, yung ating plumule derived and inflorescence derived that we were able to field plant at ARC, ano yun, bumubunga na ngayon. Marahil at the seedling stage, yung morphologically, may sa anatomy ng seedlings, magkakaroon niya ng difference kasi grown sa laboratory, yung isa, harden na talaga sa screen house. So yung mga sa number ng stomata, yung mga ganun, magkakaroon niya ng difference. But yung sa pag naka-adapt na sila doon sa labas, katulad nga nung na field plant, so far, parehas lang ang nila. Ang isang may pagkakaiba, yung sa makapuno. Katulad ang makapuno, pag yung natural na makapuno ang ating itinanim sa field, no? Yung, di ba, sabi natin, yung kabuwig, yung may puno ng makapuno na bumubunga, but they are giving as much as 25% makapuno. Kasi ang kinukuha natin doon, yung normal nut, na kinuha natin doon sa puno na bumubunga ng makapuno. Kasi makapuno per se, hindi siya mag-germinate sa seed nuts, no? So, magkakaroon yun ng difference kasi yung makapuno na embryo culture, true to type na ang makapuno yon, magbibigay na siya ng at least 75 na kaya niya magbunga ng 100% makapuno, lalo na kung nasa isolated plantation siya. Pero yun nga, katulad nung nabanggit ko kanina, yung sa case namin sa Albay, nasa gitna siya ng iba-ibang klase ng nyog, and yet we are getting 80%. So, ayun, yung doon sa aspetong yun sa makapuno. Nare-rescue natin yung makapuno, and still, na-rescue mo na, magbibigay pa siya ng mas mataas na makapuno yield. Ma'am Seda, may dadagdag sa tanong niya? Wala. Yung sa anatomy niya at the seedling stage, lalo sa plantlets, yung sa physiology niya at the lab stage, meron yung makikitang pagkakaiba. Sino pa?
Other more question? Parang gusto niyo pang makinig, ha? It's really amazing na ganito yung resulta nung ating ginawa natin. Kasi they were really doing it before na with limited funds. But they said, nung nagbuhos ng pondo, mas nakita nila na marami silang napoproduce kasi this technology is really very expensive. Yung mga chemicals talagang piling-pili. And then, kahit yung activated carbon, meron talagang brand na dapat yun ang gagamitin. And kaya most of our budget doon na papunta sa mga different ingredients para doon sa media dito. So, wala na po? Sir.
Miss Dr. Cueto, anong kailangan for you to expand your work? What are the obstacles in what you're doing? Para we can find solutions. Sabihin mo na anong problema.
Gusto ko po marahil sasagot niyan yung mga kasama ko sa program. Sino? Miss Anis. Para marinig natin yung nasa kanilang Kasi yung sa amin, yung problema, ganito pa rin, year in, year out. So, we might as well hear from them. Doc Ann.
Totoo lang, sa case ng laboratory namin sa UP Mindanao, we are already doing very well dun sa colloid production. But we are restricted, syempre, because we have limited number of people, staff, laboratory staff, funding, we can own, with the funding that is given, we can only process about 500 embryos to extract the plumules per month. No? Medyo nahihirapan pa kami kasi ilan lang yung sa lab ay 6 full-time staff. So, yun. So, oo. If there are more funds, then we can expand the laboratory, hire more people, because that will be exponential to the increase in number of possible plantlets that can be produced.
Tao ang ano. Tao yung gagawa.
So that we can encourage the young people in the audience to enter into these things. But we want to know what the real issues are. Anybody? Magsabi na ng totoo.
Siguro po skilled and dedicated staff who will do the inoculation work. Yun lang po. Talagang pagmamahal sa trabaho sa tissue culture. Napakahirap po magawa ng trabahong ito. Sa totoo lang po.
Nobody has said anything about how difficult it is to procure materials. I think that's an endemic problem in the university. So if those are the real problem, you have to identify the problems. I've talked with so many researchers and they say, look, I know when I went to the Senate, sabi ng senator, PCA used to have a budget of 5 billion. And you could not spend the money. That's why in 1 year, the budget went from 5 billion to 1.8 because you couldn't spend the money fast enough. On the other hand, the COA treats researchers like they were dishonest people. So anytime na maghingi ka ng pahirapan ka, padaan ka sa butas ng karayom, di ba? So in other words, there are structural problems that you face. Now, I'm trying to raise that issue because we had a conference with new UP President Danicon, and that's a problem in the university, sa UP, yung procurement. But you have to look at those issues to find a solution. And I think Danicon has some ideas. So maybe you could meet with people like President Concepcion and see how you can, the process is fixed because right now, yung researchers, hirap na hirap kayo talaga You want something and then by the time you pass through COA, sometimes the impression I get is that COA treats everybody as though they were dishonest. And researchers actually have to go to the COA to have it fixed. But if that is the issue, kailangan collectively, we should work for it and sabihin natin, hey, how are we gonna go ahead if we have this problem? How are we gonna attract young people to come join us? Anyway, I'd like to surface the problems kasi.
Very good, sir. Kasi hindi naman ang problema na. Totoo yun, sir. Kasi for the chemicals, example, for the chemicals, we would be requiring yung Sigma brand or Merck brand. Pero bawal maglagay ng brand. So what will be coming yung kung anong brands na hindi naman tested pa. So talagang may problema doon sa, example lang yung sa chemicals.
So anyway, you ask the heads to, hey, these are the real problems. Tell us.
Alam naman nila, sir.
But like si Danicon has solutions.
Okay. Very good. That's good. So yung mga taga-UP system. Estong.
Nabanggit ni sir yung procurement. Talagang yun ang problema, sir. Totoo lang. Lalo na yung potassium nitrate. Ngayon problema namin. Ang namin permit, parang 1 kilo lang. So, ngayon nahirapan kami kasi pag hindi dumating sa oras yun, talagang wala, yung mga cultures masira. So, hanggang ngayon, dito pa kami sa Manila nag-apply ng permit, renewable pa. So, ewan ko lang kung mahanapan ito ng solusyon. Lalo na sa Zamboanga, maigpit sila kasi makakalain nila gagawing bomba So, pag ma-justify sana ito, yun nahirapan namin. Itong may mga clearance pa o police na permit. Yan lang, sir. Yung potassium nitrate.
Yun, hindi lang sa procurement. We have to deal with PNP for the licenses. Nanggaling kami doon just yesterday and this morning, nandun si Boots. Akala ko, nung pumunta ako 2 weeks ago, akala ko, ang problema ko lang ay NBI clearance para makakuha ng maka-apply ng license as purchaser. Ngayong naka-apply na si Boots, kailangan pa pala ng isang permit to purchase. Iba pala yung purchaser at saka to purchase. Magkaiba pa na ganun. Kaya medyo complicated. Not only yung procurement, pero yung mga specific requirements para for certain chemicals, yung ganun. Tapos sa laboratory, halimbawa, mag-equip tayo, kailangan naman na i-upgrade mo, hindi naman pala pwede na 2-phase lang yung linya mo, kailangan pala 3-phase. Maglagay ka ng medyo upgraded na equipment, magsasuffer naman yung ibang equipment kasi yung supply natin ng kuryente sa ngayon, ewan ko sa amin, talagang underrated. Ang Zamboanga lalo na. Talagang grabe ang how erratic yung supply ng kuryente. Yung mga ganung bagay. Kaya lang, mga bagay-bagay na beyond our control ang nangyayari doon. Kaya settled sa genset. Yun.
Sir, tsaka yung tao nitong panay project-based kailangan may support talaga yung mga institution na kaya nilang magbigay ng tao. Kasi nga sabi, bago mo ma-acquire ang skills, ang tagal. Tapos pag natapos ang project, wala na yung taong tinrain mo for so long. Babalik ka ulit sa iilang tao. Paano tayo makakakuha nito? Hindi naman baka humingi tayo ng commitment sa funding agency na ang tao nandun for matagal na panahon. Kasi nga ay to train, 3 taon na kami nasa training stage pa lang So paano yon lalo na kung once we have this going, alam na, meron na. Tapos na yung funding. We get another funding. Hindi naman continuing to agad-agad. So, yung papasok ulit, back to zero na naman. So, yun ang isa sa malaking problema pa rin, sir. Ang sustainability ng tao.
The point I was trying to raise is that we all have to work together because I don't know if you're aware of it, but there's a worldwide war against science. A lot of people nowadays really are anti-science. Donald Trump sa US is a case in point that they are reducing monies for environmental control and so all of that. The point is we have to bring our concerns to the right people and we have to help ourselves. In the last election, I was so disappointed na meron tayong party for scientists, but in some areas, nasisiro tayo. We have to work amongst ourselves and convince other people that we all have to work together to uplift science because in the final analysis, it's only technological change which will change our lives. Di ba? But we have to work together. And then, we have to work with lawmakers. We have to work with head ng PCA, the Department of Agriculture, and even President Duterte says he's pro-modernizing us. But we have to have our voices held. Yung mga young people specifically, I want you to become very vocal and say, hey, what's in our future for us? But you have to raise your voices, and young voices are heard if you just think about it, and you can influence a lot of other people. So again, I want to reiterate, there is a war against science. People don't, if you look at National Geographic, there was a cover story in, I think, March 2012. People don't believe that men went to the moon. People think that immunization will cause some mental diseases. They think biotechnology is evil, which we all know it's not true. Because biotechnology will change our lives. And there are new technologies now which will help us. Right now, the biotechnology we're doing is 1990s technology. There's a new technology called CRISPR-Cas9. You don't have to get transferred genes from our organism to the next. You can do it there. So the regulations regarding that will be different from the old technology. And me, personally, I'm trying to create a biotech law. Because our judicial system, hindi ba, sa BT Talong,
they stopped it and then unanimously brought it back, but they made it more complicated. Yun namang administrative, pwede namang restraining order. So ang labanan na lang natin is to create laws. And we can do it. If you talk to your congressman and you talk to proper people, but we have to be organized. Yun lang po. Salamat.
Ayaw nyo pa yan? Meron tayong extra Thank you very, very much, Dr. Ben Pecson. Siya yung aming sa biotechnology talaga. And I know meron kayong isasagawa o ipapapasang law. You will try to work it out. So, sasama tayo dyan? Sasama ba tayo sa kanya? Okay. Last words from you?
Ayun, thank you everyone for coming, and I would like to take this opportunity to personally thank my advisors, Dr. Laude and Dr. Tony, for continuing believing in me. Isa sila sa mga inspirasyon ko sa trabahong to.
Okay. Ako naman, papasalamat din kami ng marami kay Dr. Christy Cueto for pursuing this. And of course, our project leaders dito sa CISET, kahit malalayo yung pinanggalingan nila, nag-attend dito from Mindanao, from Bicol, from Zamboanga, General Bato is there. Actually, lahat ng aming embryos galing sa Zamboanga. We have almost 70 or 80,000, 100 plus na pala. Kung yun ay ginawa nating seednut, meron na tayong 100,000 seedlings from seednut. Pero, pinupursu pa rin namin to dahil pag umandar ang technology na to, mas marami tayong mapuproduce at matutulungan. This is really to augment yung ating requirement. I think we need 120 million planting materials for this year. Actually, we have been cracking our head in computing, sir. Yung tanong mo kanina, nag-compute na rin kami diyan kung ilan at gaano. Kasi pinepeg namin ang aming mapuproduce per plumule nung una is 10,000. And then, nung dumating dito yung si Dr. Oropesa, are you not crazy? Sabi ganun niya. Parang nahihilo siya doon sa aming mga projections. Kaya, tinarget namin ang 1,000, pero nung dumating yung aming bagong secretary, si Secretary de la Peña, bakit nyo pipilitin ang sarili nyo kung hindi pa naman nyo talaga makukuha yon? So, palaging doon kayo i-measure doon sa inyong target na yon. So, yun na yun. Sinabi ni Dr. Christy, na kung 200 ngayon per plumule, but we will work for more. Ang importante, yung repeatability ng technology. So although this takes about 3 to 4 years bago makakome up tayo ng plantlet, once na magdire-diretso to, padami to ng padami. Exponential talaga ang growth kasi mas masipag kang mag-transfer ng mga colloid, mas maraming somatic embryos ang mapoform into plantlets and so on and so forth. So, siguro yung in terms of the sustainability of funding, after 5 years, may 2 years pa kasi kami, sabi naman nila exponential din daw yung magiging pondo ng R&D according to the DOST. With our harmonized R&D agenda, we are really expecting for exponential amount of funds for R&D. And syempre, isa to sa major technologies na Pinoy na rin ang ating sasabihin kasi nakita ko doon, kasama niyo rin si Carlos Oropesa. Sumaling tama si Sir Lorena na nung pumunta siya dito sa Pilipinas, may nakita din silang technique na hindi niyo nakita. Kaya ayaw niyang sabihin hanggat hindi tayo nagbabayad sa kanya ng royalty. Pero palagi ko nasabi na rin niya yung mga technology na yon. Kasi kung nakikinig kami sa kanya and then tinatry nila, umuubra. Yung iba. But to tell you, this is really a very expensive technology. But kung ito'y mag-work, malaking may tutulong to sa ating coconut industry. So I would like, at this point, we will be presenting here a certificate of appreciation to our speaker. I will read the citation. The Philippine Council for Agriculture, Aquatic and Natural Resources Research and Development, PCAARRD, under the Department of Science and Technology, presents this certificate of appreciation. Please come over. One-man show kasi ito. To Dr. Cristeta A. Cueto for serving as resource person during the Technology Forum on Coconut Somatic Embryogenesis Technology conducted on July 13, 2017 as part of the National Science and Technology Week celebration given this 13th day of July 2017 at the Philippine Training Center, PTTC, Pasay City. Signed, Reynaldo B. Iborra, our Acting Executive Director. Ha? Andun yung ano niya. So, thank you very much for bearing with us. So, the snacks for everyone. By the way, ako nga po pala si Edna Anit. I forgot to introduce myself at the start. And our rapporteur is Miss Alisa Carol Ibarra. And I don't know whether if Miss Christine Panaligan is over there. She is also one of our staff who work with me dito sa Somatic Embryogenesis Technology Program. So thank you very much for your active participation and your attendance here. Your snacks will be served downstairs. So thank you very much.